My dear students, in this class, let us try to understand the laws proposed by Gregor Johann Mendel in the field of genetics. And the laws proposed by Gregor Johann Mendel are known as Mendel laws of inheritance. And why Mendel has become so successful and how could he arrive at a wonderful conclusion of inheritance. All these things can be learned in today's class. Success is a formula, and Mendel exactly followed that success formula. And chance also favored him like anything. And before understanding this permutations and combinations proposed by Gregor Johann Mendel, let us try to recollect some of the terms used in genetics. Monohybrid cross is the cross where only a single character is taken into account. Dihybrid cross is a cross where two characters are considered during the process of experimentation. Phenotype is the physical appearance of an organism, whereas genotype is the genetic makeup of an organism. And coming to the reasons for the success of Mendel, Mendel selected Pisum sativum plant, garden pea plant by chance. And this pea plant, this garden pea plant, this Pisum sativum plant is an annual plant. As a result, Mendel could study many generations within less time. He studied the entire generations for nearly 9 consecutive years. And apart from that, he maintained the record of qualitative and quantitative data. And he knows mathematics very well. And he could interpret the data in a scientific and logical manner. And surprisingly, self-pollination is also possible in pea plant, and also cross-pollination is also possible in pea plant. Self-pollination means if the pollen grains of the flower falls on the stigma of the same flower in the same plant, that is called self-pollination. Self-pollination may be induced, and if it's not warranted, if Mendel wanted to go for cross-pollination, then he prevented the self-pollination by covering the stigma of the flowers with polythene cover. That means Gregor Mendel performed controlled pollinations. This is another reason for the success of Mendel. And he did not rush up, he was not in a hurry. He studied only 7 pairs of contrasting characters in the garden pea plant. And apart from that, all the 7 pairs of contrasting characters selected by Mendel are controlled by different set of chromosomes. This is the place where chance favored Gregor Mendel. He selected only 7 pairs of contrasting characters, and all these 7 pairs of contrasting characters are controlled by different sets of chromosomes within the garden pea plant. So because of all these reasons, Gregor Mendel became successful, and he was regarded as father of genetics. So let us see all these factors. Mendel worked on Pisum sativum plant. Garden pea plant is an annual plant. Mendel studied 7 pairs of contrasting characters. He studied only a single or a pair of characters at a time. That means he performed either monohybrid cross or dihybrid cross. And self-pollination is induced if needed, and self-pollination is prevented, and cross-pollination is induced if needed. So he performed controlled crosses. He made controlled crosses and kept careful numerical records of the results. Mendel concentrated only on one character at a time. He kept accurate records, both qualitative record and quantitative record. And he used statistical tools, methods for analyzing the results. The characters selected by Mendel surprisingly were present on different chromosomes. All the 7 pairs of contrasting traits selected by him showed complete dominance. This is another chance. And here you can see the various characters studied by Gregor Mendel. One is a seed shape, which has got 2 variations, namely round and wrinkled, seed color, which has got two versions, namely yellow and green, pod shape, inflated and constricted, pod color green and yellow, flower color purple and white, flower location axial and terminal, plant size tall or short. Out of these 7 pairs of contrasting characters, he studied either one or two at a time. He performed either monohybrid cross or dihybrid cross. Based on the monohybrid cross and dihybrid cross, he arrived at certain conclusions and he formulated certain principles, certain theories, which were known as Mendel's laws of inheritance. So let us take monohybrid cross into consideration. So here let us take the plant size into consideration and let us repeat the experiment done by Gregor Mendel. So monohybrid cross involves a single character. In this slide you can see on one side there is pure tall plant and on the other side there is pure dwarf plant. Pure tall plant is represented by capital T, that is homozygous tall plant, and pure dwarf plant is represented by small t, that is homozygous dwarf plant. And this generation is called parental generation. This generation is called parental generation. So here you can see this is capital T, pure tall plant. This is small t. This is pure dwarf plant. This is homozygous tall. This is homozygous dwarf. And this generation is called parental generation. So what is parental generation? Parental generation includes the plants that belong to the parents. So there are two parents. One is pure or homozygous tall plant and another one is pure or homozygous dwarf plant. And if these two plants are crossed with each other, then F1 generation appears. In the F1 generation, you observe the plants that are formed are all tall in nature. Externally, they are all tall plants in F1 generation. But this is heterozygous tall plant. This is heterozygous tall plant. What is heterozygous tall plant means? You observe here, this is in the parental generation, this is pure tall plant. This is homozygous tall plant where two alleles are similar, that is capital T. Whereas in F1 generation, the tall plant that is formed is phenotypically tall, but genotypically it is not homozygous, it is heterozygous. So this is heterozygous. So like that, the F1 generation received both genes for tallness and also for dwarfness, but it appears to be tall. Genetic makeup is different. It's not pure. Genetic makeup is different, but phenotypically it appears to be tall in nature. So this condition is called dominance. So here let me say, in F1 generation, this generation is called F1 generation. In F1 generation, the character that appears is called a dominant character and the character that is not seen is called a recessive character. So the character that is exhibited in F1 generation, that becomes patent in F1 generation, is called a dominant character. And the character that is not expressed in F1 generation is called a recessive character. In this, you can see in this heterozygous tall plant that is formed in the F1 generation, the tallness is expressed, whereas the dwarfness is masked, the dwarfness is not seen, but still it is there in the form of gene. And this is called a recessive character. So law of dominance states that based on this, Gregor Mendel proposed one theory called law of dominance. Law of dominance states that the character that appears in F1 generation is a dominant character and the character that is not seen, that is masked, that is hidden, that is suppressed in F1 generation is called recessive character. And the dominant character is expressed in both homozygous condition and also heterozygous condition, whereas recessive character is always expressed in homozygous condition. So that is about the law of dominance in F1 generation. Now let us go to F2 generation. F2 generation, you can see in F2 generation, these two genes are separated and they reach the gametes individually. This is called law of segregation. So, law of segregation, in law of segregation, you can see capital T is also received in one gamete and small t is received in some other gamete. That means these are segregated individually into the gametes. This is called law of segregation. So, here you can see the gametes, on this side capital T is there, and on this side not capital T small t is there, and here capital T small t is there. Now, let us see, this is capital T, this is small t, this is capital T, this is small t. So, this is all these genes or all these alleles are received from this capital T small t plant in F1 generation. And if capital T combines with capital T is formed. And if capital T combines with small t small t is formed. And if small t combines with capital T small t is formed. If small t combines with small t is formed. And this square, you can also draw a square like this, and this is called Punnett square. This square is called Punnett square. If you observe this Punnett square, 4 plants are formed in F2 generation. 4 types of plants are formed in F2 generation. Out of these 4 plants, 3 are taller. This one, and this one. Capital T is taller. Capital T small t is also taller. That means there are 3 tall plants in F2 generation. There is only one dwarf plant in F2 generation. As a result, phenotypic ratio in F2 generation is 3:1. There are 3 tall plants visibly and 1 dwarf plant visibly. But if you understand the genotype, if you study the genotype, there is one homozygous tall plant, 2 heterozygous tall plants, only one homozygous dwarf plant, and this is called genotypic ratio, that is 1:2:1. So, like that Mendel calculated phenotypic and genotypic ratios in the monohybrid cross in the F2 generation. Phenotypic ratio in the monohybrid cross is 3:1 and genotypic ratio is 1:2:1. And he proposed 2 laws, namely law of dominance and law of segregation. And there is another theory called law of independent assortment. We can take that example in dihybrid cross later. So, that is about the monohybrid experimentation performed by Gregor John Mendel. And this is law of dominance. The character that appears in F1 generation is called a dominant character. Dominant character is expressed in both homozygous and heterozygous conditions. Recessive character is expressed only in homozygous condition. Law of segregation states that when 2 traits come together in one hybrid pair, the 2 characters do not mix with each other and are independent of each other. Each gamete receives one of the 2 alleles during meiosis of the chromosome. So, that is about today's topic. Again tomorrow we will meet to discuss dihybrid cross and its phenotypic and genotypic ratios. So, let me make a review of the today's lesson. Gregor John Mendel is father of genetics. He experimented with Pisum sativum or G.L.N pea plant. He studied 7 pairs of contrasting characters and he applied statistical tools to these experiments. Monohybrid cross is a cross where a single character is considered. Dihybrid cross is a cross where 2 characters are considered. In monohybrid cross, there are 3 generations under the observation of Gregor Mendel, namely parental generation, F1 generation, F2 generation. F1 generation, only dominant plants appeared. The character that appeared in F1 generation is called a dominant character, and that is not seen in F1 generation is called a recessive character. And in F2 generation, phenotypic ratio is 3:1 and genotypic ratio is 1:2:1. And the square that is used to represent the different plants and its population is called Punnett square. So that is about today's lesson. Thank you very much.